In our last video, we talked about what enzymes are and how they work by different mechanisms and some examples of enzymes that you need to know. So in this particular video, we'll think about some of the factors that affect their activity. And the first one is a classic one, which is temperature. So if you increase the temperature, what you're doing is increasing the thermal energy in the environment. So what that means is that the molecules, like the enzyme and the substrate, will absorb that thermal energy and convert that into kinetic energy. So that means that the molecules can now move quicker. That means that there is a higher chance for the active site of the enzymes and the substrate to collide with one another, and therefore increasing the rates of reaction. When it comes to talk about factors affecting enzyme activity, you will often see a lot of graphs, and you do need to know what these graphs mean and be able to interpret them. So let's say on the x-axis here, we're doing an experiment where we are changing the temperature in which the molecules are in. And on this side, we have the rates of reaction. When it's very cold, like 0 degrees Celsius, there's obviously not a lot of reaction happening. As we slowly increase the temperature, the rate is going to increase as well, and reach a point where we say that it's the maximum, so we call that the optimum temperature. But very quickly after we pass the optimum temperature, it will quickly decrease like that. It's important to make sure that you get the terms right. When there is a very low temperature, we say that the enzymes are inactive. What that means is that both the enzyme and the substrate, they don't have a lot of kinetic energy, so they're moving very slowly, and therefore there will be less collisions happening, so the rate is quite low. We get to our optimum temperature, which is the best part because everything's just happening very quickly. So when we say that we reach a maximum rate of reaction, and we call that Vmax. If we get past the optimum temperature and keep going up the temperature, the rate will fall down very, very quickly, because at that point, enzymes are denatured. This is a very important word to know. When an enzyme is denatured, we say that the active site is destroyed. By definition, denaturation is the loss of active site. If we have an enzyme that looks like this, and that V shape there is the active site, when we increase the temperature, it will become denatured, so it will look something like this. Originally having a very clear V shape, now we can no longer have that V shape active site, so the substrate can no longer fit into the active site, and therefore no reaction happens. In exams, often they will probably give you a graph like that and then ask you to interpret it, quoting data from the graph or from a table, or perhaps they will give you data and that you need to sketch the graph and explain what's happening. Some key bits that people often will make mistake on is to say that when it's too cold, the enzymes become denatured. When it's too cold, it just means that they're not moving quickly, but it doesn't mean that the shape is lost. But when it gets too hot, it definitely is denatured, because they're moving, they have so much kinetic energy that the atoms move apart much further than they should do, breaking the bonds that hold the tertiary structure together, and in that sense you will then break the active site, and that's kind of the explanation for it. There's a particular quantity term that you need to know, which is called Q10. The number of Q10 represents how the rate increases with every 10 degrees Celsius increase. So for example, if we say Q10 equals 2, that means the rate is doubled every 10 degrees Celsius increase. If we say Q10 is 3, it means the rate triples with every 10 degrees Celsius increase. So it really depends on what it is. Now the second factor you need to know is pH. What pH stands for is the sort of partial concentration of hydrogen ions in a particular solution. So we say the higher the pH, it means there's a low proton or hydrogen ion concentration. It means that it's alkaline, and vice versa. If we have a low pH, it means that we have a high proton concentration, and therefore it's acidic. If you go back to one of our previous videos, you will see that we have different protein structure levels. There is a particular bit, for example, in the tertiary structure, which is the 3D shape of the protein. It's held together by different types of bonds, and some of them are like covalent bonds or ionic bonds, disulfide bridges, hydrogen bonds, hydrophobic interactions, that sort of stuff. pH, like we mentioned earlier, it relates to the proton concentration, and these protons can then affect the protein structure. And because we say pH, it correlates to proton concentration, we say that they affect two particular bonds, which are ionic bonds and also hydrogen bonds. Let's say we have an enzyme that looks like this, and it will normally fit a substrate that looks like that. And in that particular situation, we got just a few protons floating about, let's say maybe 5 of them. And that enzyme is perfectly happy. It will react with the substrate to do whatever chemical reaction it needs to do. However, if we put in a lot more protons into the solution, it's going to start changing the structure a little bit, so it might turn into something that looks like this. So it's got lots and lots of protons around it, and what they do is that they interact with the polar and charged R groups of amino acids, and that's how they affect the ionic bonds and the hydrogen bonds that are formed. As you can see, there was a complete loss of active site as well, so the substrate of this particular shape can no longer fit into that particular active site there. And let's say if in another situation where we take away some of the protons in the solution, so it might change the shape into something that looks like this. In this particular case, we have a lot less protons, but because we say that the protons actually interact with the amino acids to hold the protein shape, with the some of the protons gone, they can no longer have that interaction, and therefore the shape is lost. So in this bit, that active site is now too flat to react with the substrate, so it can no longer bind to it either, so it will not work. Again, you can see this is where the rate has peaked, so once again that's what we called Vmax, or it's the maximum rate, and this is the optimum pH. As you can see here, the proton concentration will really affect the structure of the enzyme, and therefore we say in both of these cases, here and here, they are still both denatured, which is different from in temperature when we say it's inactive if it's too cold, whereas it's only denatured if it's too hot. Now obviously this is a very generic graph, you probably have realized. We set the optimum pH here as 7, however in reality different enzymes will have different optimum pHs. So for example, we can have pepsin, which is a protease in the stomach, its optimum pH will be pH 2. Whereas trypsin, which is another protease, but this time it exists in the intestines, they have a optimum pH of 8. So depending on the enzyme and also where they work, they would have a different optimum pH. So it's very important to remember that it's not just a generic graph for all enzymes. And then we come to the last one, which is simple enough, substrate or enzyme concentration. This is a really easy one, doesn't take much to understand this. If you have more substrate, obviously the rate is going to increase because there's more things to be broken down. If you've got more enzymes, it means you've got more factories to process the substrate, so the rate also increases. However, we say that this occurs only to a certain extent because one of them will start becoming limiting. So if I show you as, again, with a graph. So this time, let's say we have our substrate concentration on the x-axis and the rate on the y-axis. In the very beginning, if we have no substrate at all, it's going to be 0, of course. As we increase the concentration of substrate, it's going to increase the rate because there's actually something to be broken down. So we increase. If we say that the enzyme concentration stays the same, eventually it's going to plateau. Like we mentioned before, this is when we say that it has reached its maximum rate, Vmax. And the reason for that is because at Vmax, we say that all of the enzymes are occupied. So that means there's no longer any active site really available at that point. So the enzymes are basically working at maximum capacity, so therefore it reaches a maximum rate. Number one, all of the active sites are occupied. It's good to use some keywords as well, so another way of saying it is to say the maximum concentration of enzymes that are complex has been formed. The enzymes are working at maximum capacity, we can no longer increase the rate. The only way to do that is to add in more enzymes so that we can push it further up before it plateaus again. And this graph will be the same if we change this particular bit to enzyme concentration instead. And that's pretty much it. So we got our temperature, we've got pH, and we've got substrate or enzyme concentration, how they affect the rates of reaction of enzymes.